start.
Thank you very much, first of all, for the opportunity to participate in this exciting meeting, and also for the introduction. And today, as you already announced, I'm going to talk about the opportunity to change, to stimulate neuroplasticity in neurodegenerative diseases using one of the signaling molecules in our body, ghrelin. Along with the increase in life expectancy over the last century, however, comes the increased risk for development of age-related disorders, acute or chronic, which include metabolic and neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, Huntington disease. These chronic conditions share some main characteristics such as impaired synaptic activity and plasticity, reduction of autophagy, loss of neurons in motor and sensory cognitive systems, which lead to cognitive and motor decline. And also they have in common the strong correlation between the metabolic changes and neurodegeneration. Dementia is one of the signs of those neurodegenerative diseases, and seems that it takes epidemic dimensions. If we look at the data from the Alzheimer's Disease Organization and the World Health Organization, every 3 seconds someone in the world develops dementia. This is enormous burden for the society because, as we can see, the financial cost for the last year it was 1.3 trillion, but in 2030 it's expected to be doubled. There is also exponential growth in the number of people with dementia. In 2019, 55 million have it, and in 2050, the middle of the century, is expected almost 140 million people to be demented. It's important to distinguish dementia and Alzheimer's disease. Dementia is a general term for decline in mental ability, severe enough to interfere with the daily life. And Alzheimer's is the most common cause of dementia, and we can see it in 60–80% of the dementia cases. Alzheimer's is a specific disease, while dementia is not, it's just a symptom. The types of dementia which progress and are irreversible, unfortunately, because there is a big time frame between the onset of the disease, the damage in the brain, and the appearance of the first symptoms. Sometimes it's approximately 1 or 2 decades. Dementia describes a group of symptoms which are associated with a decline in memory, reasoning or other thinking skills. It also affects the behavior, feelings and emotions of the people. And it's caused by damage of brain cells which affect their ability to communicate. Many different conditions cause dementia, and if we look at this data here, you can see that the biggest cause for dementia, this is the Alzheimer's disease, 60–70% of the people, followed by the vascular dementia in 10–20% of the cases. Then we have frontotemporal dementia, 10%, dementia with Lewy bodies, 4%, and a younger onset of Alzheimer's in approximately 1% of the people. But there are also other bit more rare diseases like Creutzfeldt-Jakob disease, Huntington disease, alcohol-related dementia is also found, and there is a mixed type of dementia when the brain changes are due to more than one type of affection simultaneously. We can find different pathological changes in the brain of those people. First of all, we have neuronal damage, which is characterized by a beta-amyloid protein, neurofibrillary tangles. There is a genetic predisposition for those changes, and some areas are extremely sensitive, vulnerable. There are also brain lesions in the cortex, which give us opportunity to divide the development of Alzheimer's disease into 6 stages based on the changes across the cerebral cortex. A big role in the development of Alzheimer is also the change in the extracellular matrix molecules, those recognition and adhesion molecules. And what is observed by many research groups, this is the elevated levels of certain components of the extracellular matrix, particularly L1 molecule. There is, of course, as a result of all these changes, impaired neuronal connectivity and activity, which leads to abnormal neuronal activation. Sometimes Alzheimer's disease is accompanied, includes epileptic seizures also. I'm not going into too many details about the genetic aspects of the Alzheimer's disease, which lead to complex brain changes, but I have to mention that the greatest known risk factor for Alzheimer's disease is the advanced age. However, Alzheimer's disease is not a normal part of the aging. Supporting that statement, we have to look at the statistics. Approximately 200,000 Americans under the age of 65 are having young onset Alzheimer's disease, which starts at age, the brain damage starts at age of 30, 50, and those people are approximately 5% of the Alzheimer patients. There are several rare genes which directly cause Alzheimer's disease. Those are the so-called familial Alzheimer's disease, and many family members in multiple generations are affected. Here belong two groups of genes. This is first, this is those are the risk genes, particularly apolipoprotein E, a POI gene on chromosome 19, which is responsible for the 40–60% of the patients developing Alzheimer's disease, and deterministic genes which are reasoning for development of Alzheimer in 1 or less % of the cases. The genes are divided also into early onset. These are PS1, 2, APP, and also late onset POI genes. And in addition to the genetic factors, there are also epigenetic factors. In these cases, we have sporadic development of Alzheimer's disease, which is due to different toxic substances, exposure to them, and also infectious diseases. There are very rare cases, and here you can see the PET scan of the brain of a patient, which is quite unique because what you see here, this is presenilin-1 mutation carrier brain, who, however, doesn't have early onset of the Alzheimer's disease. She's 71, 72 years old, but she didn't show these early symptoms, which should have appeared 20, 30 years ago. This lady has 2 copies of this poetry, Christchurch homozygote, and as you can see here from the image, it's unusually high level of amyloid plaques on this upper part. The more red is the color, the bigger is the quantity of those plaques. However, she has a limited tau protein, as you can see here, and also neurodegeneration is very low, particularly this memory decline, which is not typical for her age. The changes in the brain include this formation of senile plaques, extracellular deposits of beta amyloid protein. However, this amyloid beta protein helps memory consolidation when there is a normal functioning and metabolism in the brain. Practically, this is before the beta amyloid protein impairs the memory. The toxic form of beta amyloid is the soluble version of the protein, and also a second morphological change in the Alzheimer brain, as you can see here on this specimen from the hippocampus of a patient. These are the neurofibrillary tangles, which are aggregates of hyperphosphorylated tau protein. These changes are due to mutations in the amyloid precursor protein gene, which induces a cascade of events leading to the neuronal cell deaths. As part of those cascades, as I already mentioned, this is the hyperphosphorylation of this tau protein, which is manifestation of beta amyloid cascades, and it leads to formation of neurofibrillary tangles, which you see here, and in parallel with these changes, the amyloid precursor protein gene mutation leads to erroneous beta amyloid protein processing, and as a result, we have beta amyloid depositions and formation of these senile plaques. These changes are not innocent because, as we can see here, they affect the synaptic functioning in the brain. Soluble beta amyloid dimers and oligomers, which are formed during the beta amyloid aggregations, they are toxic because they have preferences towards repeatedly active neurons, and they block the glutamate reuptake by the presynaptic terminals and also of the neurons and also the astrocytes. And this leads to hyper activation and eventually neurodegeneration of those brains. There is no specific therapy for dementia and the different neurodegenerative diseases which cause it, because unfortunately the symptoms appeared quite late after the onset of the brain damage. But nevertheless, those attempts to modify the disease, because we can't cure it, they have to address the risk genes which mediate cholesterol and lipid regulation, synaptic network functioning, microgliosis and neuroinflammation, which are due to decreased glia-mediated clearance of beta amyloid. Also, this apolipoprotein E4, which promotes the tau-mediated neurodegeneration and leads to glial activation. Fortunately, lately there are some molecules, there is some hope based on the immune reactions. The immunotherapy is quite promising because they're already available on the market, some amyloid beta monoclonal antibodies, which give some successful effect. They lead to at least to improvement of the functioning of the brain. Actually, the basis for memory consolidation, this is the neuronal cell plasticity. And the neuronal plasticity, as most of you already know, this is the ability to alter the neuronal response to environmental stimuli. It is a basic property of the nerve system, which ensures both the brain normal functioning and its capacity to partially regenerate after development of different diseases. Plasticity encompasses both synaptogenesis during the early development and also the compensatory and corrective plasticity in the adult brain. How we could counteract the complexity of the nerve system injury during acute or chronic neurodegenerative diseases. The regeneration after injury requires activation of a cohort of cellular and extracellular cascades triggered by molecules with essential role in the development. Practically, the regeneration, brain regeneration, repeats more or less the developmental stages. So we have to look for molecules which are capable of initiating neurogenesis, not only the neurogenic niches like the supraventricular zone and the hippocampus, but also in other brain areas. We have to look for molecules which can reorganize the synaptic contacts which are impaired, as you already saw during those morphological changes, and they also orchestrate remodeling of the extracellular matrix. Synaptic efficacy and plasticity are the basis for learning and memory consolidation, and neurodegenerative diseases impair network connectivity and activity, which has as a consequence deterioration of cognitive abilities. However, quite a lot of studies found also a decreased level of ghrelin, which is neuropeptide and gastric hormone. They found it decreased in the plasma of the patients or experimental animals. And that molecule caught our attention, and we decided to see actually how we could change the neuronal and synaptic plasticity using ghrelin. Is ghrelin a method of choice to treat these neurodegenerative diseases? This is a very small molecule, 28 amino acid. It's produced, initially discovered by Kojima in 1999 in the stomach, but later on it was found also in many other organs like pancreas, kidneys, testis, placenta. It's found in the hypothalamus, arcuate nucleus, and the periventricular nucleus, but the data about the presence of ghrelin, expression of ghrelin in the brain are quite contradictory. There is a triad which is evolutionarily conserved, and this is the ghrelin receptor, which is the growth hormone secretagogue receptor 1. Also the GOAT, ghrelin O-acetyltransferase, which is catalyzing ghrelin octanoylation enzyme, incredibly important. And as you can see here, ghrelin intervenes with the regulation of almost all functions of all organ systems in our body. Ghrelin caught our attention particularly because of his role in the functioning of the nerve system. It regulates not only the food intake, but it also orchestrates high brain functions like sleep in wakefulness, memory consolidation, etc. I'm not going to give details about this scheme because it's quite detailed, but it actually shows what are the major functions of ghrelin, which activates the ghrelin receptor, transmembrane protein, and triggers different cascades in the cells. It has a cytoprotective effect because it increases the autophagy, which is down-regulated in neurodegenerative diseases. It increases the beta oxidation and cell viability, and reduces the apoptosis. Ghrelin also triggers the proliferation and prevents the formations of scar in the intercellular space. And what we are working at the moment, our research group, this is the role of ghrelin into the morphogenesis, because here there are a lot of questions which has to be answered. What is interesting is that ghrelin not only activates the growth hormone secretagogue receptor, but it also leads to the internalization of this receptor, and also the translocation of the receptor into the nucleus, and probably that's how it is able to interfere with morphogenesis, to stimulate morphogenesis. Some years ago, before we decided to check what are the effects of the ghrelin on the development of the nerve system and its activity, we first needed to be sure that the newly developing neurons, what you see here, those are dissociated neuronal cultures from cortex of newborn rats, do they express the receptor? And we followed the development of the receptor for a period of 4 weeks. At the end of the 3rd week, the neurons are considered already as mature and fully developed. So quantitative analysis shows that approximately 77% of the neurons on average express the receptor during the entire period of experiments. This gave us the courage to try to see how ghrelin could affect the neuroplasticity. So we wanted to see what is the effect of ghrelin on development of synaptic contacts. For this reason, we have control groups which were not stimulated with the ghrelin, and experimental group stimulated with ghrelin since day 1 when the cultures were seeded. And as you compare the images obtained after immunohistochemical staining for one of the synaptic markers, synaptophysin, ghrelin treated cultures develop much more synaptic contacts compared to the controls. This is week 4, and as you can see, enormous load of synapses on the surface. We used different doses and found that this effect is dose dependent. However, after 4 weeks, the dose practically different doses did not matter anymore. What we have tried and we continue to use further, this was the concentration of 0.5 micromolars, which is a bit higher than the physiological dose level of ghrelin in the circulating plasma, but this concentration has been used also from other research groups, and for consistency in our experiments, we continue to use it further. When we saw that ghrelin stimulates the synaptic formation, we tried to see what is the effect of ghrelin when we chronically treat those developing cultures. We made recordings with the multi-electrode arrays, and that's how the seated neurons look like under the microscope around the electrodes, and we were able to make recordings of the neuronal activity. There is no synaptic contact between the neurons before the end of the first week. Then the synaptogenesis starts, and this experiment, the samples you see here of those recordings are made at day 6. This is the control culture, and as you can see here, there is almost a complete silence at that period. Don't be surprised. There are 2 electrodes which show very strong activity, but in the cultures usually they are so-called pacemaker neurons, which are absolutely necessary for constantly generating action potentials which stimulate the other neurons to develop and to form synaptic contacts. At the same time, if you look at the ghrelin-treated culture, which was incubated constantly in ghrelin, you can see absolutely mature pattern of neuronal activity, which is quite unusual. I would like to point at something else. What you see here in the control culture, day 7, this pattern of activity represents more or less epileptic status. So there is a long period of silence and then collective burst, while at the day 6, the functioning of the ghrelin-treated cultures is more dispersed and at a lower level. There are no seizures. So to suppress the neuronal activity of epileptic patients looks like is not the right approach, because to function normally, neurons need some level of activity. From these experiments, we can conclude that the chronic application of ghrelin largely increases synaptogenesis, and ghrelin upregulates synapse formation via another unidentified yet activity-independent manner. So it's not due to the fact that ghrelin is a mild excitatory neurotransmitter. There must be another mechanism, and that's what we are looking for. This effect of ghrelin on synapse formation and function provide a molecular target in developing novel therapeutics for disorders which are related to neurodegeneration and impaired synaptic plasticity. So encouraged by those results, we decided to see what will be the effect of ghrelin if we put the neurons under stress. So we made in vitro model of stroke and checked what is the level of synaptic contacts between the neurons. This is before the onset of the hypoxia. Then neurons were exposed to 6 hours hypoxia, and as you can see, it reduced enormously the amount of synapses between the cells. After the restoration of oxygen supply, the synaptic contacts start to form again, but much lower level, compared to the pre-hypoxic stage. However, those cultures which were treated for 24 hours after the hypoxia with ghrelin, they showed a much higher number of synaptic contacts between the neurons, which exceeds also the level before the hypoxia. What is the probable mechanism of these effects? Ghrelin is the only known ligand for this growth hormone secretagogue receptor, which has multi membrane domains, and the majority of the effects of ghrelin are mediated via this receptor. While ghrelin is not that much expressed in the brain, the ghrelin receptor is highly expressed in the entire nerve system. And what is very important is the internalization of the ghrelin receptor and transmission to the nucleus. As a proof of principle that the effects of ghrelin are mediated via this receptor, we applied calcium imaging on dissociated dorsal root ganglia neurons. And here you can see the recording of this action potential, which is generated. You can see also the firing of those neurons when ghrelin is applied, and after washing it, this is the depolarization, which is stimulated by potassium application. To prove that the receptor is the one mediating the effects, we applied antibody against the receptor, and there is at the moment of application there was no any reaction in the neurons. Then we applied ghrelin, and there was also no discharge of those neurons. The washing out and depolarization stimulation of potassium showed that those neurons are not dead, they're vivid and active. Then we applied hypoxia to different specimens, cell cultures, and acute cerebral cortex slices to see what happened to the receptor, how it reacts when the neurons are under different pathological conditions. This is a staining which shows the expression of the receptor in those cerebral slices, and we used also a marker for neurons. This is a calretinin, and as you can see here, when we merge, this is the DAPI staining, counter-staining of the nuclei. Multipolar calretinin neurons are homogeneously distributed across the tissues. Hypoxia increases the expression of receptor. It's very clearly seen here, and ghrelin, when it applied later, brings the expression level almost to normal. This is the statistical analysis. So this we interpret, this increased level of receptor, we interpret as a compensatory mechanism for the lower ghrelin level in the plasma. When we use dissociated granule neurons from cerebellum, we also saw that hypoxia increases enormously the expression of the receptor. And here the neurons are unipolar, and they are developed mainly in clusters, and the receptor is concentrated predominantly into the nucleus. This concentration location of the receptor in the nucleus we confirmed with gold nanoparticles labeling the antibody against the receptor. And as you can see here, the hypoxia increases the expression of the receptor in the nucleus, but we can see also it on the membrane of the neurons and in some vesicles. Ghrelin application brings it back to normal compared to the pre-hypoxic normoxic stage. So recently, a lot of studies showed that ghrelin could trigger adult neurogenesis, which is very important for the repairment of the brain after acute traumas or diseases like stroke, and also in chronic neurodegenerative diseases. As you can see here, this receptor is G protein couple receptor, which triggers neurogenesis, and this is probably because it activates being addressed to the nuclei. It probably triggers different transcription factors which stimulate neurogenesis. We tried to prove that ghrelin activates adult neurogenesis, and for that purpose we used newly plated cultures. At the moment of their plating, we applied ghrelin and then measured the expression of different markers like double cortin, which is involved in the migration of newly generated immature neurons, and it's marker for potential adult neurogenesis. As you can see here, those cultures, they have a lot of precursor cells which express double cortin. We expose those cultures also to hypoxia 7 days later because it gave a chance the double cortin to be expressed. And as you can see, before the exposure to hypoxia, there is a certain level of expression of the marker. Hypoxia reduces it significantly, while exposure to ghrelin increases enormously the expression of double cortin on the neural precursor cells. ZBTB is another transcription factor which regulates the post-mitotic acquisition of area identities. It's a proliferation marker which orchestrates the neurogenesis of neurocortical projection neurons and plays a crucial role in the transition between neurogenesis and gliogenesis in cortical glial radioglial progenitor cells at the perinatal stage. Practically, in vivo, ZBTB is not expressed in the cortex after the day 7 postnatal, and we wanted to see what actually will be the effect of this of ghrelin on its expression. And this is before the hypoxia exposure. Hypoxia stimulated triggered the neurogenesis. However, it was much higher when ghrelin was applied. Here you can see the statistical analysis of this expression. Another marker for proliferating cells, this is the key 67. This marker is protein is expressed on the nuclei exclusively of proliferating cells. It's considered as a marker for progenitor cells, and during mitosis, what you can see here, it covers the surface of the chromosomes and represents 1/3 of the protein mass of the chromosomes. It prevents the adhesion of the chromosomes and guarantees the symmetric split of the chromosomes between the daughter cells. The half-life is very short. It's approximately 1 hour. And what you can see here, surprisingly, Ki67 expressed in a neuron, 3 weeks old neuron, and this NUN is a marker for developed mature neurons. It triggers division mitosis in those cells. When we applied the same experimental conditions before the hypoxia which reduces the expression of Ki67, and ghrelin exposure leads to significant increase, double more than the early stages of development and before the onset of hypoxia. Here you can see double labeling, which shows that ghrelin Ki67 is expressed not only in glial progenitors, which are labeled with marker for astrocytes, GFAP, but also in non-glial cells. Those are the neuronal progenitor cells. Much more convincing is even this one. 3 weeks later, this Ki67 is well expressed, as you can see here, the mitosis in glial progenitors. PAX6 is a marker for proliferating neurons, precursor cells. It's expressed in GFAP positive cells, glial progenitors, and also in 30% of the neuronal progenitors we can find it also. The same experiment, carotene is marked for neurons, and as you can see here, hypoxia significantly reduces the expression of PAX6 in the cortex. This is unlike the other brain areas, while ghrelin stimulation increase significantly the PAX6 expression. This is quite interesting finding because PAX6 is indicator for proliferation. So the expression of ghrelin receptor is enhanced after the hypoxia. It could have an amplifier-like effect for low concentration of ghrelin. Ghrelin receptor is present in and translocated to the nucleus and associated with the chromatin, which might affect the expression of transcription factors, which you already saw. And the hypoxia reduces the PAX6 levels in vitro. However, ghrelin upregulates it, which is quite optimistic finding. We can speculate that the ghrelin functions as a cofactor modulating PAX6 expression, possibly affecting neural progenitor cell fate or cell cycle. Ghrelin also elevates the expression of the other transcription factors and markers for proliferation, like double protein Ki67 ZBTB, and all these findings make us emphasizing the relevance of additional studies on the regulatory role of ghrelin and corroborates its therapeutic potential to ameliorate recovery after acute or chronic brain injury. At the end, I would like to acknowledge the contribution of my colleagues, Professor Lutz, Professor Milena Milosevic, who made the calcium imaging, and Andriy, who is a PhD student of Dr. Lutz. And with this, I would like to thank you for your attention.